Here are the notes for Chapter 12, Section 3, dealing with RNA and protein synthesis. There's lots of vocabulary terms in this section, so I would pause this, take a minute, make sure that you get these in your notes, that you highlight these. Again, there's a lot of vocabulary terms, and you're going to need to make sure that you know them. Those from 12.3 include gene, messenger RNA, ribosomal RNA, transfer RNA, RNA polymerase, promoter, introns, exons, codons, translation, and anticodon. Our objectives for this unit include determining the difference between RNA and DNA, understanding the three different types of RNA, describing transcription and the editing of RNA, identifying the genetic code, summarizing translation, and explaining the relationship between genes and proteins. So that's kind of where we're actually going to start. Again, we know that all of our genetic information is on genes stored in the form of DNA. Genes are the coded DNA instructions that control the productions of proteins within a cell. What does that mean? And how does that help us get from knowing our genotype going to our phenotype? How does this work? Well, proteins are often enzymes which will catalyze and regulate different chemical reactions. And the phenotypes are the results of the role that the enzymes are actually going to play. So for example, there might be an enzyme that will produce a specific pigment, and if this protein is coded for that, if we create this protein that will then become this enzyme, the plant might have a specific color. Also, if this protein makes an enzyme that produces a specific red blood cell surface antigen, it'll determine your blood type. So all of this genetic information, all of these instructions go to make these proteins, which then go on and make enzymes and help catalyze different chemical reactions. So from all of this information, we're getting different reactions that are occurring with the body, and that's where we get this kind of expression of the gene. So where does RNA fit in all of this? Well, DNA, reminder, it's in the nucleus, and it can't leave the nucleus, otherwise it could be very easily damaged. So we use RNA as the usable copy of the DNA to send those messages outside the nucleus to create those proteins. And reminder of where some of those proteins are made with the ribosomes, that type of thing. So we need to get the DNA somehow out of the nucleus and into the rest of the cell where we can synthesize these proteins. So a little bit about RNA. RNA is a long chain of nucleotides, very similar to DNA, but there's three main differences. Make sure that you write these down, that you know these. The sugar in RNA is ribose instead of deoxyribose, hence RNA versus DNA. RNA is single-stranded instead of a double-stranded. So remember, we have our double helix, kind of twisted ladder structure for DNA. RNA is just a single strand. RNA contains uracil instead of thymine. So how thymine is complementary to adenine, RNA is going to have the uracil instead of that. Now looking at RNA, again, the most important job of RNA is to create these proteins, which will then go on and influence the workings of the cell and the expression of those genes. The entire process of turning amino acids into proteins is actually controlled by the RNA. So that's why we're going to spend some time about it, looking at it. So this whole kind of central idea is we've got this DNA that has this coded instructions, and we're going to use this RNA to kind of help get those coded instructions out of the nucleus and into the rest of the cell, working to create these proteins, which will then go on and express the genetic information. There's three different types of RNA that we are going to focus on. Messenger RNA, which is often denoted as lowercase m RNA, ribosomal RNA, and transfer RNA. Messenger RNA is the one that's going to carry the copies of the DNA so that the proteins can be made. And messenger RNA, they're just messengers. They're going to send that information to the ribosome, which is going to create that protein. We have the ribosomal RNA, which along with the proteins, the RNA make up the ribosomes. So we have this ribosomal RNA along with it that are going to help actually create more of these proteins. And the transfer RNA is used during the construction of the protein, and it transfers the amino acid to the ribosome to the extended protein chain. And I've often heard transfer RNA called translation RNA because it kind of translates this information, genetic information, into the amino acids and the proteins. Again, if we take a little look here, you can look at the different types of our RNA. Our messenger RNA, which is going to be number 1 here, that's going to kind of carry that information, genetic information. We have our RNA that's involved with in the ribosomes themselves, in the production of the protein, and we have our transfer RNA, which is down here in the middle. So again, we're just going to go back and focus that there's 3 main types, and they each serve a little bit different, have a little bit different function as to what they need to do. So, okay, where does this RNA come from? There's 2 processes involved with RNA, transcription and translation. And the first one is how we get our RNA from our DNA. Remember, RNA, we need to get this transfer of the genetic information out of the nucleus and into the cell where it can be used. So this first part, transcription, is going to help do that. Translation is where we actually kind of decode some of that information, and that will be part of the video notes for the second half of section 3. So transcription, it occurs in the nucleus. Again, this whole component piece of trying to get information out of the nucleus and into the ribosomes outside, we need some way to do that. It's a similar process to how DNA replicates, but a reminder, we only have a single strand of RNA, so it needs to act a little bit different. So we're trying to take this DNA and create our RNA. And instead of using the DNA polymerase that is going to unzip that strand, the double helix, we have RNA polymerase. And what this does is it binds to a section of DNA and uses one strand as kind of a template from which the nucleotides are assembled into a strand of RNA. So if we look at this transcription here, we have our kind of RNA polymerase, which you can't see in here. But what it is doing is, again, is we're taking, kind of unwinding, unzipping this and filling in our information. So as our replication. So unlike the DNA polymerase, which does it kind of for both sides, we're just going to have this RNA polymerase just work kind of on the one side. And again, we have this uracil instead of thymine that's going to be inserted there. And that's kind of the overall gist of what happens in transcription. So remember, we're transcribing from the genetic information from DNA into RNA. The other piece of this is RNA polymerase can only bind to the DNA strand at certain points, and these points we call the promoters, which have a specific base sequence. So the promoters are where we're going to kind of start, but there's also a certain spot where we're going to stop. So you can kind of think of it as this green light, red light of the DNA strand. So again, this RNA is only going to occur during specific spots, and the promoters is that beginning of that spot. And they have a specific kind of base sequence that's going to tell the DNA polymerase that it can start there. Again, here is just another visual of this again. And as we notice, we have the DNA is going to unwind. We have our RNA, our polymerase, it's the one that's kind of starting this, and then we end up with coming out with a strand of RNA. And notice our DNA is going to wind back again after we've kind of copied that one piece. So we're almost kind of done with this idea of transcription. We have a few more pieces in here. Okay, so we have this RNA being produced from DNA, but not all of the genetic information it will code for specific proteins. So RNA is going to kind of take out some of those specific stuff. Introns are the portion of the DNA that are not expressed as an mRNA strand. So because this does not create the protein, they don't need that genetic information. The exons are the portion of the DNA that does do that. The codon, which we're going to practice a little bit more in class, is a sequence of 3 nucleotides that will determine the amino acid to be added. Okay, what does this mean? We just went through this idea of how a gene is transcribed. We get from the DNA to the RNA. Well, we just have to kind of look at, there's some segments on the RNA strand that we don't necessarily need. From this DNA, we don't need all of this information. You can kind of think of it as like an editor going in and making, small changes. If you think of someone placing an ad in the newspaper, words are money. So what the RNA undergoes is trying to get out some of this information that is not necessary. So we have our introns in the blue here, that genetic information that does not code for the protein, and the yellow pieces, which are the exons that will. So the exons are what are going to be transcribed. We're going to kind of splice all of the exons into this. So we get our new mRNA, which only contains the information we need to synthesize the protein. So you get this kind of finished mRNA. And that's kind of where we're going to leave off for here. This last little bit of the first part of this involves codons, which we talked about before as being this sequence of 3 nucleotides that will actually determine the amino acid that is going to be added. But we were going to do that section in class. So if you're not going to be in class, when we go over this, make sure you take a look in the book at page 302 and 303 to look at how we use that codon information to synthesize our proteins. Here ends the video notes for Chapter 12, Section 3, Part 1.